So now we have Annemieke Aartsma, who's going to talk about gene therapy, what the genetic therapies look like, what our future looks like. And I think you met Annemieke, who serves us chocolate and now cookies this morning, which is exactly what we need to refuel. So thank you again. Thank you again for coming, for Nate and for Rachel, and for your wonderful words to start off this morning. And now, Annemieke, it's you. Are you mic'd?
Thank you very much, Pat. And I think so, coming back to what Rachel was saying, because so I'm a researcher, so I talk to patients and parents only if I come to meetings like this. And well, this is my first patient meeting in over 2.5 years. And I think if I could narrow it down to one word, it's inspiring. And I think I didn't realize how much I missed that inspiration and motivation until I came here and talked to you all again and met all the wonderful parents and patients. So I've been asked to talk about the future of genetic therapies. And I think, well, we had the quote from Einstein yesterday about thinking about the problem and the solution. So if we look to the future, we have to also look to the past and the present to understand where the problems are and then why we need to go into a certain direction. Also, some of this you may have heard already in other talks. I think indeed it's overwhelming. There's a lot of science. It's very complicated. So I'm going to repeat certain things and then, well, hopefully at some point it will click if you hear it the first, second, third, fourth, whatever time. So apologies if there is some overlap. Hopefully it will be useful. You heard about dystrophin and that Duchenne patients don't make dystrophin. And in healthy muscles, dystrophin prevents the muscles from being damaged when it's used. So lacking dystrophin, patients chronically experience damage. And that leads to chronic inflammation. That leads to formation of scar tissue. And it leads also to an impaired regeneration process, and gradually that leads to loss of muscle tissue and loss of muscle function. Don't have to explain the consequences of that because you have patients or you are a patient, so you know your child, you know yourself. But of course, so all of this is caused by not having dystrophin, and then the logical solution is, well, what if we restore dystrophin? If we restore dystrophin, then all these problems should go away. And there's one thing to bear in mind, and that is that you don't need dystrophin for muscle to exist. You need dystrophin to prevent it from going away. And that means that if you restore dystrophin, you won't undo the damage that has accumulated. You will only prevent further damage from occurring or slow down the further damage. And I know that that's not a very hopeful message, but it's really good that you understand this so that you have the right expectations from these genetic therapies. So how can we restore dystrophin? We have different tools. We have gene therapy, you heard about it yesterday. We have exon skipping, you heard about it yesterday. We have genome editing. It's not been presented about, but I'm sure you heard about it. CRISPR-Cas, on the news, on the internet, everywhere. So I'll explain for each. So how does this work? Where are we now and what are the challenges? And of course the challenges are linked to the future direction because, well, based on the challenges, we plan and strategize for the future. So we'll start with gene therapy and we'll go over them one by one. So Duchenne patients cannot make dystrophin because the gene that codes for this protein is mutated. So what if we give patients a functional copy of the gene? Then with that copy they can make dystrophin. Well, to deliver that gene, to deliver that DNA, you need a delivery vehicle. And the only thing that currently can deliver a DNA to muscle is the AAV, the adeno-associated virus. And this is a really small virus. If you were in the gene therapy session yesterday, you will have seen the small dots on Tim Kripe's slide. It's really small, and that is why it can deliver things to muscle. If it was bigger, it wouldn't be able to reach it. However, it's a virus. So that means that if you've seen AAV before, your body, when you get the gene therapy, will recognize it. And we heard yesterday also that, well, you get 400 times more viruses than you have red blood cells. You get really a lot of viruses. So if your body has seen this virus in the past, because, well, it also is a natural virus, there will be a big immune response. It's not just that it will negate the virus, the gene therapy from doing its work. There will be a huge immune response, and that will have dangerous consequences. So this is why it's important, if you do gene therapy, that the patient hasn't seen this AAV before. I mentioned already it's small, that's why it can reach muscle, but because it's small, the dystrophin gene, which happens to be huge, doesn't fit. So we need to have a minimalist approach. We need to make it as short as possible so that it fits. Now there's 3 components that have to be in these, what we call viral vectors. So we need some viral sequences because, well, the micro-dystrophin needs to know it has to go into these capsids that you see on the top right. You need the promoter. This is a volume switch. The gene needs to be on in muscle and in heart. And you need the code for the micro-dystrophin, and it's micro-dystrophin because the whole dystrophin doesn't work. So there's really a minimalist approach, and at the bottom in blue you see the schematic, scientific depiction of the dystrophin gene, and below that an analog that I like to use. This is a carabiner, which you use if you, for instance, go rock climbing, and this carabiner has 2 crucial domains. It clicks to your harness and it clicks to the rope. Well, dystrophin does something similar. It connects the skeleton of the muscle fibers to the connective tissue surrounding the muscle fibers, and in that way it provides stability. In the middle, you can see there's, well, this elastic domain, and that's very similar. Dystrophin has this elastic domain, 24 repeats and 4 hinges. Now, what they've done with the micro-dystrophin, they've taken these 2 crucial domains, so these domains that connect, and then they've shortened the elastic part. So you can see there's, well, depending on which company, there's between 2 or 3 hinges, and there's 4 or 5 of these repeat units. So it's really small, but these crucial domains binding to the skeleton of your muscle fiber and the connective tissue surrounding it, they are there. So it can make this connection. Now, currently, there's trials ongoing. You've heard about it yesterday. Solid, Pfizer, Sarepta, Roche, and also in Europe, there's a Genethon Institute is also running a clinical trial. And they use different viral subtypes. We call them serotypes, so 9, 7.4, 8. And because patients will be at risk for a really dangerous immune response if they've seen the virus before they're being treated, patients with antibodies to whatever vector the companies are using are excluded from the clinical trials. Pat gave a rough estimate. She said, well, I think about 100 patients have been treated so far. That was also my rough estimate when I made my slides. And I see it says patients in Dutch, so that's a nice, a nice autocorrect there. But we estimate that roughly 100 patients by now have been treated, and we have some general findings from that. So what we know is that it is possible. With AAV, you can, if you do intravenous delivery, you can reach the muscles. And I think for those of us who've been in the field for longer, that really is an amazing accomplishment, and that has taken years to develop to reach this point. However, the levels vary. So for some doses and for some AAVs, we see 5% of the fibers are dystrophin positive. For some, we see 80%. And then also the total levels can be from around 1% to almost 100%. So this varies. And not just between different viruses and different microdystrophins, but also within trials between different patients. Side effects have been seen as well. So they are severe to very severe, and sadly, 1 patient so far has passed away due to the side effects. So I don't want to trivialize them. And they are related to the dose. So the higher the dose is, the more patients are at risk of having severe side effects. Unfortunately, the higher the dose is, the more microdystrophin you will express. So if you use a lower dose, you're at less risk for side effects, but the dystrophin levels will also be lower and less fibers will express the microdystrophin. Probably the side effects are also age-related. The older you are, the more mature your immune system is, and the more it will react to the virus. And of course, in this case, this is bad. You don't want the body to react to the virus, but this is what the immune system does. Viruses normally are bad news, so it makes perfect sense that the body responds to it. It doesn't know that this time the virus has good intentions. So what's interesting about the side effects is that there's two types. There's the acute side effects. You heard about them yesterday in the gene therapy session. There's the nausea, the vomiting, liver damage, kidney failure, sepsis, death. It sounds horrible, and it is. But there's also another type of side effect, and that occurs later. That is the muscle breakdown. Rhabdomyolysis is the jargon term. Myocarditis, inflammation of the heart. And we know now that this is due to an immune response to the micro-dystrophin. So not to the virus, but to the micro-dystrophin. And it's only seen in patients who have a large deletion at the start of the gene. So how does this come? And I will say, so kudos to the companies who work on this, because once they realized that this was happening, they all talked to each other, academics connected them. They've been very open, they've been very collaborative in trying to mitigate everything. So that's really good, but we don't know for sure what is happening, so I'll just give you my personal speculation. So what we know is that the immune system reacts to foreign proteins, so proteins that are unfamiliar to it. Duchenne patients, we always say, don't make dystrophin, but that's not really actually true. So why doesn't the micro-dystrophin trigger immune response in all? Well, that is because many patients do make some dystrophins. And if we again go back to this big carabiner dystrophin, that's the dystrophin you have in skeletal muscle and in brain. But there's also shorter dystrophins, for instance in brain, that only contain the last bit, so the last part of the carabiner. That is made in almost all patients. So only if you have a mutation really late in the gene, you're unable to make this dystrophin. So most patients will make this dystrophin, and that will not be foreign. So that part of the microdystrophin will be known to most patients. The first part of the dystrophin, so most patients have a mutation somewhere in the middle of that elastic domain. That means the first part is produced. It's not functional, it's not stable, but it's produced, broken down, but the immune system will have seen it. The immune system doesn't know whether it's functional or not, it just knows, okay, this is something that we, this body, make. So for most patients, that part is also not new, but something that they are familiar with. The exception is the patients that have big deletions at the beginning of the gene because they cannot make this protein. And now suddenly they get the microdystrophin where this part of the protein is present. And they say, well, we've never seen this. This must be a foreign invader. We need to react. So that's why patients with a large deletion at the beginning of the gene have this immune response, and that's also why now they are excluded from clinical trials. And of course, the challenge is, why do some patients do this and others not? We don't know that yet, but for now, for safety reasons, all patients at risk are excluded. So there's more challenges. And the first one is, how functional are these microdystrophins? They're really small, they contain the crucial domains, but they're not going to be fully functional. They're smaller, they're much smaller than the dystrophins that Becker patients make. And we know they're functional in mice and dogs. But you see here the nice picture of the mice standing on its hind legs. That's not how the mouse normally moves. The mouse is a four-legged creature. So that has consequences to the strain that is put on its muscle compared to us walking on two legs. So how functional are they? We don't know. The placebo-controlled trials are ongoing. Then the second question is, how long will these microdystrophins persist? They are not fully functional. We know that for sure. We know in Becker patients, there's also turnover of muscle. So with time, there will be muscle turnover and this microdystrophin will be lost. And what we don't know is, will that take 5 years, 10 years, 20, 50? We don't know that. But we do know that, well, when it happens, if you want to retreat, well, for now you have a problem because, well, once you've treated the patient with 400 times the number of erythrocytes of AAV viruses, for sure there will be an immune response. For sure the body will have recognized it. So this is something that has to be solved. So how can we make sure that, if needed, we can retreat patients? And also, how can we treat patients who have seen the virus before, just say in its natural environment, and who are now excluded from the trials and the treatment? And then the problem about the immune response to the microdystrophin, well, we need to understand better why some patients do this and why others don't, because that hopefully can help us to counteract it. For now, these patients are excluded from the trials. But it may also have consequences for restoring the full-length dystrophin, because the full-length dystrophin will have parts for every patient. It will have parts that are foreign. And then the question is, are these parts, will they trigger the immune response or not? Because maybe certain parts are better at that than others. So there's a lot that we don't know yet and that we need to study. So what's going on? What are people working on? Well, first of all, they're trying to optimize the AAV viruses. So if we can generate a virus that goes to muscle better, because we keep saying, well, it goes to muscle, but it goes also to other tissues. It goes to liver, and that's why a lot of the problems occur. If we can make sure it goes better to muscle, you need a lower dose, so there's less safety concerns. Can we use something else instead of a virus? And lipid nanoparticles are often mentioned. So these are lipids, so the fats, that you can complex with the DNA or with RNA and then deliver. So far for muscles, this doesn't work very well, and that's again because these nanoparticles, well, they say nano, small, but they're actually quite big. They're much larger than the AAV. So for now, for muscle, they don't work very well. They work really well for liver. So whenever something has a lipid, the liver will like it and take it out of the bloodstream. And I mentioned already, for repeated delivery, people are thinking about this as well. So with the lipid nanoparticles, it would be possible to repeat the treatment if it was needed. But also, if you have a lot of different AAVs, maybe you treat first with type 1 and then with type 2, and that might work. But this is really work in progress. So that is the current situation and the future directions of the gene therapy. We're going to move now to exon skipping, which is something I've been working on for almost 22 years. And I'll very briefly explain the basics again for those of you who were not at the exon skip session yesterday or who didn't understand it, which, well, I mean, it took me a while to get it as well. So don't feel bad about that. So what we have here is the genetic code of the dystrophin protein. And the genetic code is actually dispersed over the gene. And we call these pieces of genetic code exons. And in between are pieces that don't contain code, and they're called introns. Now, before the protein can be translated from the code, these exons need to be linked together. And you see here an example of a patient who misses exon 48, 49, and 50. And you also see the shape 47 doesn't fit to 51. That means the code is disrupted. And that means when the translation happens, everything goes well until exon 47, and then in exon 51, the code is disrupted, so translation stops. What we can see is 47 would fit to 52. So what we want when the cell is linking all these exons together, we want it to ignore exon 51. Now, how can we do that? We do that with antisense oligonucleotides, AONs, also called ASOs, oligos, PMOs, all the same thing. It binds specifically, in this case, to exon 51. It hides it from the machinery that connects all these exons together. And now 47 is linked to 52. And patients can produce a Becker-like protein. Now, the thing is, these effects are not permanent. So it's not acting on the DNA, it's acting on the temporary RNA copy. And that is why these exon skipping drugs need to be delivered every month or every week or every 6 weeks, depending on what type of AON is used. So challenge here is that this is a mutation-specific approach. And we had a discussion about this yesterday in the exon skipping session. Exon 51 skipping applies to the largest group, 14% of patients. So that means it doesn't apply to 86, which is more than 14%. So if you want to develop this for more patients, you need to develop AONs for all these different exons. And you can see that the percentages go down quickly. So the top 4 is, well, 8% or more, but then it becomes smaller, like 4%, 3%, 2% of patients. So that is another challenge. So where are we now? We have 4 AONs approved in the US, so exon 51, 2 for exon 53, 1 for exon 45. So this applies to about 30% of patients. However, the approval is based only on restoration of dystrophin at low levels. So less than 1% to about 5%. And the question is whether these low levels will be enough to slow down disease progression. We don't know that yet, and confirmatory studies to assess that are currently ongoing. So we don't know yet, we just know the trick works, so to say, dystrophin can be restored. But it's clear that there's room for improvement and that there's need for improvement because, well, beyond a certain point, it doesn't matter whether you make 50% of dystrophin or 80% of dystrophin, but I think everything less than 10%, it definitely matters whether you make 1%, 5%, 10%. So we need to improve this. So what are the bottlenecks? The bottlenecks are getting the AONs to the skeletal muscle and the heart. And another bottleneck is that for each exon, you need to develop a specific AON. So what are companies, researchers doing to try and mitigate that? Well, the first thing is that they link conjugates to the AONs. Again, if you were in the exon skipping session yesterday, you will have heard about arginine-rich peptides or PPMOs. Again, they're all similar things, so they're peptides that are small, that are positively charged, and that improve uptake of the AONs. It's not muscle specific, they improve uptake in all tissues. And examples of this are the SRP-5051. I think, well, it has a name now, it's eteplirsen with something in front of it, but it's basically eteplirsen with the peptide. PepGen has an exon 51 compound with their own specific peptide, and Entrada has their own specific peptide linked to, and it says here exon 45, but it should be exon 44. So they're all having their own peptide to try and improve delivery to everything, and that then includes muscle. And there are safety concerns, so far it's been reported in humans and in animals that there's hypomagnesemia, which means not enough magnesium levels in the blood and in the body. You can supplement for this, but this is really something that needs to be monitored to see, is this a controllable problem, or is this in the long term a safety risk, or are there other safety risks? So those are things that, well, the clinical trials that are now ongoing will need to clarify. Well, then you can say, well, why not use something that delivers better just to muscle? Well, people are working on that as well. So the muscle-specific uptake, you see here, the other one was just a drill. You just drill a hole in all the cells and more will be taken up. What if you address the oligo to go to muscle? And this can be done with antibodies that target the transferrin receptor, which is a protein that's highly expressed on muscle. So you add something that recognizes this protein, you add it to the oligo, and then it will go preferentially to muscle. And both Dyne and Avidity are developing this for their respective oligos. But also here there's challenges because these peptides that the others used were really small. These antibodies or antibody fragments are larger. So can there be antibodies against these antibodies? Can the immune system recognize it? Because these antibodies are not normal, they're foreign. So that's a possibility that needs to be studied. And then even though transferrin is highly expressed on muscle, it is also expressed on liver. So that's again another thing that needs to be considered. So what will go best? Will it go so well to muscle that the liver is less of a problem, or will the liver actually take up most of it? Because the liver is really good at taking up things. So you heard also from Kevin Flanagan yesterday and also on Thursday about combining exon skipping with gene therapy. So can we give an ASO gene, so to say? So you don't have to repeat the treatment, you have permanent expression of this antisense gene. And Kevin Flanagan is doing this, so the antisense gene is called U7-snRNA. You hijack the system, and instead of what it normally binds to, it now will bind to whatever you tell it. And in Kevin's case, that is exon 2 for exon 2 duplications. And he has done this now for 3 patients in an academic setting, and well, you've seen the results, especially the baby that he treated. The results look very promising. Audentes, Astellas were developing this as well, preclinically, but that development has stopped, and I don't know whether other companies may pick up and try this approach as well. All the challenges for safety and production of AAV apply here as well. But the only exception is that the dystrophins that are formed here will be much larger than the micro-dystrophin. So that is something to consider. The downside is that with micro-dystrophin, you can give it regardless of the mutation. Well, here you would have to develop a system for different mutations, just like with the AONs. And then finally, for the exon skipping part, the applicability. So we need to develop an AON for all these different mutation types, and the subgroups become really small. Can't we use a trick where we say, let's develop a mix of AONs that then apply to a larger group of patients, knowing there's a lot of deletions between exon 45 and 55, knowing that a deletion of exon 45 to 55 doesn't disrupt the code, is found in very mildly affected Becker patients. Can't we use that? So make a mix of AONs targeting 45, 46, 47, 48, until 45. And then we give that to everyone who has a mutation in that region. Well, there's challenges. The first challenge is, of course, that, well, this mix needs to reach all of the different cells, because if one component is missing, there's a challenge that it generates another skip that is not productive. But there's also a regulatory challenge, and I try to explain that with this graph here. So here you have a deletion of exon 47 to 50. If you would give the 45 to 55 mix, you would give every component in the mix, but you would also give an AON targeting 47, 48, 49, and 50. And we know the patient doesn't have these exons. So we know these AONs have no target in this patient. And the regulators really don't like that, because you're now giving something that may have side effects, while you know already, before you treat the patients, that there will be no beneficial effects. So then you would have to customize the mix for each and every patient, and of course, then we're back to square one, where you have to customize it, so you have to develop it independently. So this is really a challenge, and there's also discussions with the regulators. Well, knowing that at some point these AONs work, will it be enough to just show, look, we restore dystrophin for the exon 23 skip or the exon 33 skip or whatever. But that is for the future. So finally, genome editing. So what is genome editing? Genome editing is a way to modify the DNA, so the genetic code. And I'm sure you've seen fancy pictures, but I'll just like to explain it like this. It's a scissor that cuts your DNA, and it has a GPS guide that tells the scissor where to cut. So that's basically the component. And the scissor is called Cas9, and the GPS guide is called guide. So why would you want to cut the DNA? Because it contains the protein information. So why would you want to cut it? Well, you want to cut it to activate the DNA repair mechanism. DNA is, the body really protects DNA because it contains the code that we need to make proteins. So, and when it's cut, it will degrade very quickly. So it wants to repair this break as soon as possible. And there's different ways in which this can be done. There's the recombination, that's a copy and paste approach that happens in dividing cells. So if you give the cell a template, like, well, this part is missing, but look, this should be in there, it will copy and paste and repair the damage. However, that only happens in dividing cells. And skeletal muscle is not a dividing cell type, it's what we call post-mitotic. It stopped dividing. So there's another mechanism for those cells, and that's called non-homologous end joining, and I like to call it the glue system, because what's basically done is the two pieces are stuck together. And this will create small mutations, because part of it may have already gone. Maybe there's some DNA building blocks floating around that will be put in, like, okay, if you're here, you probably were part of this DNA. So it creates mutations, but at least the break is repaired and further damage is avoided. So that is what can be done in muscle. So if you want to correct mistakes or add what is missing for Duchenne, that is currently not possible because this recombination system, the copy-paste system, doesn't work. But you can make small errors, and you can use that to induce permanent exon skipping. So not having every week treatment, but just once. So how can you do this? Another example here, this is a patient with a deletion of exon 52. And you can see 51 doesn't fit to 53, but 50 would fit to 53. Now, rather than hiding this exon while the the cell is figuring all these pieces together. What if we cut from the DNA with 2 of these scissors? We cut before, we cut after, exon 51 is gone, and now we've restored the genetic code because now 50 can be joined to 53, and every time a copy is made of this DNA, the frame is readable and the code is readable, and a Becker-like protein can be produced. So this uses 2 scissors, but the more scissors, the more risk that the scissor cuts somewhere else, that things go wrong. Can we use 1 scissor? Yeah, we can do that as well. So the exon is recognized by the body, so that it's during the put it together process. If we cut just in front of it, the exon will no longer be recognized. So most of it will be there, but it won't be recognized. So you get the same thing. Every time a copy is made, 50 is linked to 53, and a Becker-like protein can be produced. And if you hear single cut, dual cut, well, this is the difference. They both result in the same thing. So genome editing, well, at least in the Netherlands, it's hyped. Many people think it's gonna solve everything. And also researchers, scientists who work with it are really very enthusiastic about it. And it makes sense because there's a lot of things that we can do now with genome editing that we couldn't before. So before it took years and years. If we wanted to make cell or animal models, it took a long time and some were simply not possible. Now, in a few weeks, we can make cell and animal models. So that's really helpful to study pathology, to study disease, to make mouse models, to develop therapies. So it's understandable that we researchers are excited. And then, of course, if it's that easy to make these models, you start thinking, well, can we use this as a therapy? And then suddenly things that are not possible with exon skipping, such as multi-exon skipping, are now possible with genome editing, because we just cut before exon 45 and after exon 45, and everything in between is missing, and it doesn't matter where your mutation is, you need these 2 scissors for everyone. So in theory, that would work. And people have also shown that in cell and animal models, these things are possible. There's even a clinical trial now ongoing for a liver disease that uses genome editing. But there's 2 buts, and the first but is the second bullet point. So this approach got the Nobel Prize for chemistry. It didn't get the Nobel Prize for medicine. And that's because there's not a single medicine developed with it yet. They might come, but currently they are not there. It's, for now, a tool to study diseases, to make models. No therapy is there yet. And then so it seems to be in clinical trials for a liver disease, so the step to Duchenne then is small. Well, sadly, no. Because again, we're back to the delivery problem that, well, has been a red thread of my talk, unfortunately. So because you need to deliver these components, you need to deliver the scissor, and you need to deliver the guide. And to do that, well, you need something, and again, we're back with AAV. So you need 1 AAV to deliver the scissor, because the scissor is big, so it fills 1 AAV, and you need 1 AAV to deliver the guide. And what we know is that it's not simple like, well, 1-on-1, say 1 part scissor, 1 part guide. No, you need to add more guides for some reason than scissors. So you need to add more than with the micro-dystrophin. So then the question is, are these higher doses, are they tolerable by patients? So that's just from the, say, can we do this in patients aspect? But there's also safety concerns, because if you deliver this, we were talking about longevity. Here, you don't want these scissors to be expressed long. You just want them to do their job, make the cut, and then disappear. You don't want them to float around to maybe encounter another region and then cut there as well and do who knows what harm. So you want them to be there a short time, but if you deliver them with AAV, they will be there for a long time, and that will accumulate the correct cuts, but it will also accumulate the wrong cuts. And what we've also seen with AAV, so AAV normally doesn't go into our genome, so into our DNA. However, if you start cutting up the DNA, it does. And of course, well, if you deliver something that cuts DNA, it will happen, and it has at least already been shown in animal models that this happens. So can we use another system then to deliver this? Well, yes, so these lipid nanoparticles can deliver RNA that encodes the scissor. And then you get a very short-term expression of the scissor, and then the work is done, and it's degraded, and it's no longer able to cut. So that is a much safer way. However, these lipid nanoparticles, we've seen already, there's a reason they started their clinical trial with the liver disease, because lipid nanoparticles deliver very well to liver, but not so much to muscle. So that is really something that needs to be developed. If we can manage to deliver DNA to muscle without viruses, then this becomes much safer than if we need the viruses. And then also something about the therapeutic effect to be expected. And I started with this as well. The damage that has been accumulated with the current techniques cannot be undone. The efficiency of genome editing in animal models currently is rather low. And when you read the papers, the press releases, often they will say, we corrected the mutation. But that's a misnomer, because they didn't correct it, they just made the code readable. So the patients will not produce a normal dystrophin, but a partially functional Becker-type dystrophin. So that's really, it's good to bear in mind that if they say the mutation has been corrected, they don't mean normal dystrophin will be produced. And the time of intervention will matter, as I said already. And then there's also the safety aspect. We know that the scissor doesn't only cut where it's supposed to cut, it will cut somewhere else. And once that's done, there's no undo button. It's there to stay, and it can have harmful consequences. There is, however, a new system, the base and the prime editors, and these don't cut the DNA. They are like Tipex, so you have an A, it should be a C, and this system just, well, it sees the A, and well, the guide tells it this A shouldn't be here, and it will convert the A into a C. Because there's no cuts, there's less risk of intended and unintended cuts. But for now, the efficiency of this system is really very low. And how can you use this? If you have small mistakes, you can correct them. But you can also use this to make exons invisible and again get permanent exon skipping. So what is the future here? We're very early stage with genome editing. It's preclinical. I don't expect there will be clinical trials in the near future because we need to improve things. We need to improve the specificity of the scissor. It should cut only where it's supposed to cut and not somewhere else. We need to improve the efficiency. I mentioned, well, multi-exon skipping is in theory possible, but it's very inefficient currently. So we need to improve this in order to make this work. And also the editors, the Tipex system, they're safer, but currently very inefficient. So that needs to be improved as well. And then I all the time talked about you want to deliver to all the different skeletal muscles. But something to consider is also local delivery. Whoever had the Pfizer or the Moderna vaccine knows it's possible to deliver short pieces of RNA to muscle by just direct injection. Maybe that is something, maybe we deliver the mRNA of the Cas9 with the guides, and then you get temporary effect. You will treat just one muscle, but I think there's muscles, some muscles that are really important for daily activities. If you can at least treat those, lacking treatment of the whole body, that might be something to consider. So that is the last slide. So it's possible, we know it's possible to restore dystrophin with exon skipping and AAV dystrophin. However, there's a lot of room for improvement and there's outstanding questions. Genome editing is still in the preclinical stage. I don't know if and when it ever will be in clinical stage. And I think, so I hope I've not taken away your hope because there's so much that we don't know yet, so much that needs to be optimized. But I will say that there's many dedicated scientists working in industry, working at academia, that really try to work and solve these problems. So you have a lot of bright minds working on this for you. And that was my last slide. So if there's time for questions, I'm happy to take them now plenary or you approach me. I'm still here all day, so you can also approach me during breaks. So thank you very much.